This is the first half of a lecture on bacterial transformation for April 13th, 2020. And the focus of this lecture is to really define some terms regarding transformation, talk about things that make bacteria naturally competent to perform transformation, and then outline the mechanism of transformation in a specific bacteria, S. pneumoniae. And so we previously talked about how bacteria get their genetic variation. And because they don't sexually reproduce, they use both mutation and a process known as horizontal gene transfer to really gain genetic diversity that they can use to evolve. And horizontal gene transfer is the transfer of genes from one sort of adult organism to another adult organism. And we had talked about conjugation last time, and today we're going to focus on transformation, which is the uptake of free DNA from the environment into a bacterial cell. And this DNA can be either linear, as you can see here on the right, or it can be plasmid DNA. And DNA can be found in the environment for a variety of reasons, one of which is dead or lysed cells. And so when cells lyse or die, they can release DNA into the environment. And if bacteria are capable, they can actually take up that DNA into themselves. And what's important to remember is that in transformation, the DNA that's taken in is then maintained in a heritable way through what's known as a stable transformation. And so in the case of linear DNA, as you can see here on the right, it can be taken into a bacterial cell and then either integrated into the host chromosome, which is known as the stable transformation, and then that integrated DNA would be passed down to any progeny cells generated by this bacterial cell here. But DNA can also be degraded before it's integrated into the bacterial chromosome, and that transformation would be considered unsuccessful because anything encoded on these DNA fragments here would never be expressed and also would not be able to be passed on to subsequent progeny cells. And so DNA can also be transformed into bacteria in the form of a plasmid or a circular piece of DNA. And in the case of plasmid DNA, once they're taken up into the cells, technically a stable transformation has happened because there's no integration necessary for plasmid DNA. Many plasmids can exist extrachromosomally or outside of this blue bacterial chromosome, and they can still be passed on to any progeny cells. And so not all cells can undergo the process of transformation. As you can see here, this guy in the middle is feeling a little bit incompetent because he's unable to uptake any of these free-floating DNA molecules. Because in order to take up free DNA from the environment, a cell has to be what's known as competent. And competent really just means able to take up DNA. And the idea of competency in nature, or like naturally occurring competent cells, is sort of complex. The most common factor that leads to competency is the stage of growth in which the bacteria are in. Usually during the exponential or logistic growth phase, when bacteria are multiplying rapidly, they also tend to have a high competency. But the competency of bacterial cells varies based on the species of bacteria. Some species, for example, depend on what's known as a competence factor, and so competent bacteria secrete a specific protein that can then bind to cells in the same area and upregulate expression of the proteins needed for transformation. Some species don't require a competence factor at all. And so there's a lot of diversity in what makes cells competent, but we know that competency is really important for some species, especially the ones that form biofilms or those kind of sticky conglomerates of many different microbes living together, because that's one of the ways that bacteria living in a biofilm can pass DNA information between them. And so the way that transformation works in one specific, very well-studied bacteria called S. pneumoniae is outlined in this image here. And so in the case of Escherichia, linear DNA and the external environment can bind to receptors or DNA binding proteins that are on the surface of this competent bacterial cell. And then once the linear DNA binds to receptor proteins, there are enzymes called endonucleases that can basically cut that linear piece of DNA into smaller size pieces. One strand of this double-stranded molecule will be degraded, and then that single strand can be fed into the cell through what's known as an uptake system. And so that uptake system is one of the kind of those proteins that are upregulated in response to a competence factor. And some different types of uptake systems can be seen here. And there's different diversity in what makes up an uptake system depending on which bacterium you're looking at. There is always a channel, however, where the DNA can be fed through, but the other proteins can differ between different organisms. And so once that DNA is fed through the uptake system into the competent bacterial cell, that DNA strand can align with the homologous region on the bacterial chromosome. Remember that homologous basically means same sequence. So this lac plus gene or piece of DNA that was taken into the cell will find the lac gene in the bacterial chromosome. They will align with each other, and then the external piece of DNA can be incorporated into the bacterial chromosome via a process known as homologous recombination. An enzyme named RecA is responsible for this, and so that basically consists of a cut of one strand of the bacterial chromosome out and being replaced with this new red strand that has been transformed into the bacteria. And remember that not all transformation will be linear pieces of DNA. This is a specific example from a specific species of bacteria. Often plasmid DNA is brought in through the uptake system, and it doesn't have to integrate into the genome over here, but it can exist extracellularly and still be inherited by the daughter cells after transformation of the mother. And so just to see this in animated form, you have a donor bacterium that dies and lyses, releasing pieces of DNA out into the environment, and then a competent recipient bacterium that has DNA binding proteins or receptors, which can bind to those external pieces of DNA, feed them in a single-stranded way, and then RecA can allow recombination of this homologous gene here into the donor chromosome.